Welcome to Session 2, Use of the Same Model or Modeling Strategy Across Multiple Submissions Focused on Complex Drug Products. This session is comprised of 5 talks. Drs. Andrew Babiskin and Miyoung Yoon will kick off the session with a joint presentation. Dr. Babiskin is the team lead for the locally acting PBPK team at the Division of Quantitative Methods and Modeling, the Office of Research and Standards, the Office of Generic Drugs. His current work focuses on advancing mechanism-based absorption modeling of locally acting complex products to support novel in vitro and in vivo pharmacokinetic-based methods for bioequivalence. Dr. Yoon is serving as the team lead for the quantitative clinical pharmacology team within the Division of Quantitative Methods and Modeling. She has been leading the team's efforts to conduct regulatory and research activities leveraging clinical pharmacology tools and expertise and actively developing collaborations with multiple stakeholders. Dr. James Clarke, Senior Research Scientist from Certara Simcyp Division, will deliver the second talk of the session. Dr. Clarke is specializing in mechanistic thermal absorption modeling and also works on development, enhancement, and implementation of the virtual bioequivalence module of the Simcyp simulator. Dr. Clarke will be followed by Dr. Marc, who is a Senior Manager Material Scientist at the Global Inhalation Research and Development Division at Teva. He leads the Material Sciences Group, whose primary responsibilities encompass the definition of critical materials attributes and subsequent material process controls for respiratory platform products. Maxime Merdy, PharmD, is a Senior Scientist at Simulations Plus and will give the next talk. Dr. Merdy is specializing on PBPK modeling for locally acting drug products. The last talk of this session will be delivered by Dr. Ducharme, President and CEO of Learn and Confirm and Associate Professor at the University of Montreal. Professor Ducharme has 30 years of academic, clinical, and industrial experience in pharmacometrics, infectious diseases, drug metabolism, and clinical drug and biological development. Rosu Alenga, PhD, Chemical Engineer at the Division of Quantitative Methods and Modeling, Office of Research and Standards, and Partha Roy, PhD, Director at the Office of Bioequivalence at the Office of Generic Drugs, will moderate Session 2. Dr. Walenga joined the FDA in 2015. His research interests include computational fluid dynamics modeling of orally inhaled, nasal, ophthalmic, and dermal drug products to answer questions pertaining to bioequivalence. Dr. Roy is a recognized senior clinical regulatory strategist with 22 years of drug development experience in both the US FDA and industry, involving new drugs, novel biologics, generics, and biosimilars. He currently leads an FDA CDER office that oversees the assessment of bioequivalence data required to support ANDAs. Thank you for attending Session 2.
Thank you for the introduction. My name is Andrew Babiskin, and I am opening this joint presentation between myself and Dr. Miyoung on the regulatory perspectives of modeling strategies across multiple submissions. Both of us will be providing viewpoints from our respective teams in the Division of Quantitative Methods of Modeling, or DQMM. Myself, the locally acting physiological-based pharmacokinetic, or PBK, modeling team, and Miyoung, the quantitative clinical pharmacology team, in that order. This disclaimer slide applies to both presenters and is a reminder that the views expressed here today are our own and do not represent FDA's views or policies. Before we start the discussion on model reuse or model repurposing, let's start with an initial perspective on the role of modeling simulation in both PBK and quantitative clinical pharmacology and complex generic drug product development. First, modeling simulation can be utilized to address challenges with comparative in vivo bioequivalence studies. For example, modeling simulation can aid study design, predict probability of study success, and support how study outcomes will be evaluated for BE. Modeling simulation can serve as the basis for alternative BE approaches with reduced human testing. Modeling simulation can supplement in vitro characterization-based BE approaches with model-based evaluation of in vitro study results. It can also support establishing an in vitro method as a biopredictive method. Lastly, such information gained through modeling simulation can support product-specific guidance development. Specifically, when it comes to mechanistic modeling like PBK and computational fluid dynamics, or CFD, there are a number of advantages a generic drug product development program can have when employing such a tool. By integrating all data information known about physiology, the drug substance, drug product attributes, and device parameters, if a drug-device combination, in vivo performance can be predicted at or near the site of action. Oftentimes, an in vivo study to gather such results is not feasible nor ethical. As an extension, mechanistic modeling can also be utilized within BE approaches that have reduced human testing, such as comparative clinical endpoint studies, which serve as a surrogate for drug delivery to the site of action. Implicit in the concept of model reuse is that there is a first successful regulatory use of the model. Using PBK as an example of a mechanistic model, there are a number of factors that need to be considered. First, what is the quality data being used for model development and verification and validation, or V&V, activities? For locally acting products, oftentimes the data from local tissues come from literature sources. Are the justifications made scientifically sound? Is parameter selection and optimization appropriate? Have all relevant processes been considered with special emphasis on absorption aspects for these bioequivalence approaches? Are test of reference products adequately defined in the model? How will they be compared? How well is the PK data being predicted for the product of interest, but also how well is the platform performing for a larger array of products? Lastly, we also consider when use of either virtual healthy or patient populations, or both, is appropriate. Finally, we get to the main topic of today's presentation, model reuse. So that we are thinking about this concept in the same way, I have defined model reuse in the form of this question. Can myself or any other user, such as another applicant, utilize the same modeling approach that has been successfully applied before for now a different product in a different application? It is also important to define what a platform is and what makes it distinct from the model. A PBK platform, for instance, is a system of databases and differential equations defining movement of drug through ADME processes defined by anatomy and physiology. By taking that definition, a model is an implementation of the platform parameterized by properties of the drug substance and the drug product. For my portion of this presentation, the reuse scenario is one in which the same platform will be utilized and for the same intended purpose, but the implementation or the model is different due to the product changing either in just the drug substance, or the formulation, or the dosage form as a whole. Ultimately, the aim for the model reuse is to have increased confidence on the regulatory acceptance of this modeling approach, to have more streamlined regulatory submission and a reduction in V&V activities from the applicant, and also a reduction in the reassessment of those activities from the regulatory body for which the application is submitted to. When applying the same modeling approach to different products, it is important to acknowledge that even the first use of the model, its acceptability for regulatory decision-making within applications, is supported by a history of predicting successful outcomes across multiple drug products and drug substances and commercially available platforms. The platform is what you will use to develop your product-specific model, but its credibility is independent of your specific case. Rather, we have typically considered platform credibility to be based on whether a sufficient number of diverse products can be adequately predicted given the intended purpose of the model. On the final slide of my portion of the presentation, I will cover some specific considerations, but for now I'll say that inherent in this platform performance assessment is that I should already establish a range of usage for the model beyond your product of interest. I also would like to note that while traditional V&V activities has focused on systemic exposure for oral PBK applications, for local acting products, V&V is instead focused on predictions at the site of action or administration. Provided in this slide is an example of a platform performance assessment that supported the approval of a diclofenac sodium topical gel. DQMM has published on this case, so I encourage the audience to refer to the article mentioned below for full detail. The goal of this model was to perform a virtual bioequivalence assessment of local skin tissue in the site of action, the synovial fluid, driven by the characteristics of the test and reference formulations. Given the novelty of the durable PBK modeling approach and the lack of regulatory or literature record as it comes to this model, It was important to establish the credibility of the platform beyond just the diclofenac sodium topical gel. To this end, the applicant performed a platform performance assessment by producing a variety of dermal models encompassing topical and transdermal products with diversity in both formulation and drug substance attributes and validating against both systemic and local bioavailability. This activity led to a suitably validated platform that could be utilized for the intended purpose. I'm briefly showing you Table 3 from the reference publication as a more visual depiction of what the platform performance assessment entailed, but to also further support the point that this same activity, while intended for diclofenac sodium topical gel, could apply across a variety of products and supports model reuse. I've been largely talking about model reuse and platform performance assessment in the context of PBK modeling, but let's take a look at the example with CFD modeling. While in terms of platform performance assessment, this case is not as mature as the previous example, it will be used for illustrative purposes. For this application of CFD, we are examining and predicting regional deposition of lungs after administration of an orally inhaled product. With CFD modeling, the aerosol particles that are emitted from these products after actuation are individually tracked as they transit through the mouth, throat, and lungs, where the aerosol will deposit in either of these regions or be expelled. The CFD results on the left is for a dry powder inhaler. On the right is a soft mist inhaler with different drug substances. The nature of these aerosols from these products are different, but the CFD model based on known physics and other processes can be adequately compared against in vivo data coming from gamma scintigraphy studies. The ability for a CFD model to predict a variety of aerosol types with different property lends itself to its model credibility and can establish applicability of using the model across multiple products. Now let's examine some practical considerations that should be made when deciding to reapply a model to a different case. We will return to the PBK modeling space for these considerations, but most of these concepts can apply to other types of mechanistic modeling. Probably the most important consideration is whether the intended use of the model is the same as the previous instance. Verification and validation is intricately linked to the intended purpose of the model, so it's not given that the V and V set from one modeling purpose will fully support another modeling purpose, especially considering the consequences of the decisions made based on the model. The next consideration is whether for the new product of interest is the physical, chemical, and physiological properties of the product as a whole and the drug substance consistent with the platform performance assessment previously performed. For example, if metabolism wasn't a significant consideration in the original dermal model and not part of the platform performance assessment, and the new product undergoes extensive metabolism in the skin, the PPA can be considered incomplete. Lastly, if the first use of the model also included extensive validation of local and systemic exposure for the product of interest and other products containing that drug substance, and the new product has no such information, justification of parameterization decisions may have to be supported further by more platform performance assessment activities. I thank you for your time and look forward to the panel discussion. I now hand the presentation over to my colleague, Dr. Miyoung Yoon, who will provide her perspective on reusing modeling strategies with population PK modeling.
Thank you, Dr. Babiskin, for the first part of the presentation. The second half will cover the regulatory perspectives on reusing the modeling strategies. Population PK is a data-driven approach, so it is not heavily dependent on platform or predefined model structure like PBPK models. Model Integrated Evidence Approach, or MIE, is gaining industry's interest lately, and we have seen increasing number of them in the submissions, mainly to reduce the clinical study challenges. Industry's interest on MIE is evident. What I would like to note here is the feedback about potential ways to facilitate implementation of MIE. Industry also voiced the need for standardization of MIE. We want to talk about MIE strategies from a standardization perspective. I believe we will hear details about different types and processes of MIE over the workshop today and tomorrow, so only high-level overview here. Typically includes three parts: in vivo study, population PK model, and BE study simulation. In vivo study uses alternative designs to meet the goal of each MIE type, either shorter study duration or smaller study size. Then population PK model will be developed using the real in vivo data. Then the model is used to simulate BE study using conventional BE study designs, which otherwise would have been challenging to use in the real situation. Here are graphical representations of MIEs to think about the product-specific and common across product natures of MIE, so that we think about the idea of reusing MIE processes or framework in not just for a single product, but across or multiple products. For a given type of MIE, model validation and verification would be different because in vivo data would be used for different purposes, but may be common within the specific type of MIE. This is a hypothetical example of a long-acting injectable product. So we see how study design was selected and justified for use in this in silico dosing MIE. Applicants can perform a clinical trial simulation to show the proposed approach can distinguish formulation difference with good type I error control and study power. The type I error and power comparison between the model-based study design and the conventional study shows the advantage of MIE here. In this case, significantly shorter study duration. Here, I am using the MIE framework developed on the GDUPA research program by Uppsala University to help us see what components are included in virtual BE MIE strategy. Typical process of model building using the alternative design study data, model validation, and BE studies simulation using virtual subject. As applicants are expected to follow this general framework, it can be viewed as a point of standardization. This will be discussed in more detail tomorrow by Dr. Andy Hook. Let's talk about some regulatory considerations for in silico dosing. They are based on single product proposals, but you can see these would be applicable across product. For example, type I error evaluation using clinical trial simulation processes. Again, the real in vivo study design should allow distinguishing potential difference between test and reference product in the simulated BE study. So different study designs are encouraged for testing to select most sensitive and efficient one. Standardization of MIE will also help preparing modeling analysis plan and its review. Here are some examples of regulatory considerations for virtual BE MIE. Because virtual subjects are simulated in this approach, the real in vivo study should be adequate for estimation of variability and uncertainty in formulation parameters to ensure confidence in BE conclusion. To this end, type 1 error should be evaluated before conducting virtual BE study. This shows some key regulatory considerations for population PK modeling for bioequivalence purposes. Population PK guidance has already specified the general model building and validation processes. Additional considerations should be made for BE. The model should describe formulation difference and be sensitive to detect the difference in the simulated study. Reusing means using the same modeling tools or processes used in the previous submission to meet these key regulatory considerations for a new submission, which can be multiple submissions. Standardization into a shareable platform of these processes. Again, not reusing the model itself, because population PK model, the test and reference formulation difference will still be coming from the new clinical data, so model needs to be followed the full model building and validation for each time. Now we want to get back to the point of regulatory perspective on reusing the MIE strategy. So the common benefit of MIE is clear, and we expect that its use will increase given the potential it already has shown. Therefore, it is clear the benefit of reusing the modeling strategy, most importantly for model validation and verification perspectives. To reiterate, the benefit of reusing includes help increasing the acceptability of further application of the same approach, more streamlined regulatory submission, and increase in efficiency in model validation and verification activities. Now, I would like to close the presentation on behalf of Dr. Babiskin and myself. We would like to continue discussion about fostering effective communication when same model or same modeling strategies is used across multiple submissions, and we look forward to today's panel discussion as well as tomorrow's model master file session. We would like to thank you for your attention and thank our colleagues and collaborators.
Good morning. It's a pleasure to be speaking at this workshop today. My name is James Clarke and I'm a Senior Research Scientist at SimSip. First, I'm going to talk about opportunities for reuse of mechanistic models, and later I'll focus on how we can best integrate them into the generic drug development process for complex dermatological drug products. I'd like to start by pointing out some of the advantages of mechanistic and physiologically based pharmacokinetic models. PBPK models are built from the bottom up, and they separate the systems data, i.e. the physiology, from the drug and formulation data. This information is then tied together within a mechanistic framework, most often a series of compartments and ordinary differential equations, but other types of mathematical framework can be used. A key takeaway from this slide is to recognize that the systems data and the mechanistic framework are generic to the system and are not confounded by the drug or formulation being modeled. This means they can be reused. But also, by separating these aspects, it importantly allows for mechanistic extrapolation. This can be between populations or between drugs and formulations. Here I want to take a minute to briefly talk about platform verification and open science. One advantage of using the same modeling platform for multiple projects or submissions is the accumulation of verification case studies and assessments for the platform over time. Taking the SimSip simulator as an example, we're now up to nearly 700 publications in the public realm that use this platform, with more than half of them being from independent sources such as academic institutions and pharmaceutical companies. This means anyone can go to the public literature and find a significant amount of information about the science behind the models and the vast majority of the equations on which they're built, as well as verification case studies in different areas. One thing I wanted to note also is the recent increase in publications from the dermal and biopharmaceutics areas. Historically, this graph would be heavily weighted around the bottom, with DDI and special populations being the major section. However, we can see at the top that 16% of these publications were dermal and biopharmaceutics last year, which combined makes up more than DDI or special populations, and this represents the growth in interest in these areas. For the remainder of this presentation, I'll be talking specifically about the MPML-MECDerma dermal absorption model, which is integrated within the full PBPK model of the SimSip simulator. However, the general principles I'll be discussing translate to any adequately mechanistic and well-verified PBPK model. The MPML-MECDerma has been developed over the past 8 years in collaboration with the Office of Generic Drugs through 4 grant awards, 2 of which are still ongoing. Here, when we talk about the systems data, we can look at various populations and body sites that are available, which were developed from extensive data collection from the literature. These are another reusable aspect of the model. The physiological data is integrated within a compartmental modeling approach that characterizes the different layers and phases of the epidermis and dermis. This forms a reusable, qualified and verified framework which can be combined with physicochemical properties of the drug, but also crucially for the generic drug product development, a series of mechanistic formulation models which take in vitro formulation characterization data as input. Topical drug products come in various forms and can often have multiple phases and complex dynamic processes occurring. This slide shows some of the mechanistic formulation models that can be used to capture the dynamics of multi-phase systems, such as emulsions and suspensions, and metamorphosis of these formulations when applied to the skin. The advantage of employing mechanistic models over more empirical-based approaches is the ability to utilize in vitro characterization data and build from the bottom up. This allows extrapolation and gives us the ability to perform sensitivity and safe space analysis for both referenced and test formulations. And I'll give an example of this later in the presentation. As a further note on verification, if we look specifically at the MPML-MECDerma, over the past 5 years or so, we've presented more than 20 posters at scientific conferences, and in the past couple of years, we've also published a series of publications that go into detail on the model structure and provide equations and workflows. All of these poster PDFs can be found online on the Satara website, and the journal articles are open access. Armed with these models, the next step is to think about how we can most effectively integrate mechanistic models into the generic drug development process. Depending on your background and previous experience with modeling, it's a common belief that the primary aim of developing in silico models is to replace in vitro and in vivo studies. And although it may be possible to reduce or replace some studies, that usually isn't the primary goal. In the next section, I would look briefly at the workflow for building a dermal absorption PBPK model and how we can use that to inform the development process to make the in vitro and in vivo studies more robust. A key aim of PBPK modeling is in vitro to in vivo extrapolation. In the context of dermal drug product development, that involves integrating the in vitro formulation characterization data into mechanistic formulation models. Important characteristics include drug solubility or relative affinity for various phases, drying rate, viscosity, and disperse phase or particle size if an emulsion or suspension formulation. Also, in vitro release and permeation testing can give us insights into the formulation's behavior. In the case of IVPT studies, this can be used to inform the model, and the use of IVRT studies or other similar release studies is an area of active research for us in our ongoing FDA grant projects. The developed model can then be verified using a confirm and learn process, whereby available data is used or generated to verify and optimize the model. This could include, for example, generating challenge formulations that have been deliberately altered to assess the model's ability to pick up the formulation changes. From there, we can look to use this model to extrapolate to the population of interest and simulate the question which we're asking. In this case, that would be a virtual bioequivalence assessment. An important point is this process is most effective if it takes place alongside the formulation development process. But too often the use of modeling is an afterthought only once the program has run into difficulty. By integrating mechanistic models early, they can help to reduce these issues by informing study design. On this slide, I want to quickly run through the model development process and where it can feed into the formulation development process. First of all, we need to develop a preliminary PBPK model. And this takes as input the physicochemical properties of the drug and also the reusable model that we've just discussed. From here, we can develop a systemic model, which can take, for example, in vitro metabolism data as input, and also we can use clinical data from IV or oral studies to inform and verify. Similarly, for the dermal absorption aspect of the model, we can use IVPT data or clinical dermal absorption data to inform and verify. And this brings us to a verified PBPK model for the compound. Alongside this is the formulation development process, where we can gain information on the Q1, Q2, and Q3 of the reference formulation. And this information can then feed into the PBPK model, where we can develop a preliminary model for the reference formulation. This model can be used to perform sensitivity analysis on formulation parameters, and we can identify which parameters are most sensitive and feed this back into the formulation development process. And this can inform us on the best ways to develop a test formulation. We can identify safe space for certain parameters, and that can inform how we go about the formulation development process. We can also use this information to develop challenge formulations where we deliberately alter a sensitive parameter, and we can use these challenge formulations and the models of them to verify the PBPK model later on. So we can feed this characterization data from the test and challenge formulations back into the models and form preliminary PBPK models for the test and any challenge formulations. At this point, we can move to investigative studies, for example, IVPT, IVRT, or clinical PK studies for the test, reference, and challenge formulations. And by simulating these, we can use those results to verify the PBPK model for the test and reference. Once we have verified PBPK models for the test and reference, we can utilize them to perform virtual bioequivalence assessments. If the development process was using just in vitro permeation, then at this point there would be an in vitro in vivo extrapolation step also. And we can use the test and reference verified compounds to perform a virtual bioequivalence assessment. And this gives us bioequivalence results, but also we can perform further sensitivity analysis, and from this we can define a safe space for the formulation attributes. There are various places within this workflow where we may be able to reduce or replace studies. For example, we may be able to reduce the number of investigative studies required by complementing them with a verified PBPK model, or we may be able to reduce the number of replicates required in the study. Another area is within the bioequivalence assessment itself. We've heard of the diclofenac gel example, where the virtual bioequivalence assessment was performed in place of a clinical endpoint study. But depending on the compound and formulation of interest and the specific question being asked, it may not always be appropriate to completely replace a study. However, there's still immense value in running this kind of framework alongside the development process, as there's various places where the models can be used to streamline the process. For example, the preliminary PBPK models can be used to inform investigative studies that we do need to conduct, such as estimating the LLOQ that may be required and aid in dose selection. Similarly, within the bioequivalence assessment, it's valuable to run a virtual bioequivalence assessment before conducting a practical bioequivalence study, as this can help to inform the design, such as a power analysis to estimate the required number of subjects, or to aid in dose selection, and I'll show an example of that on the later slide. But also to give a trial run of sorts that may give an opportunity to redevelop a test formulation if the model suggests it may be at risk of failure. The earlier we can identify these issues, the more value the models can bring.
Finally, I'd like to talk about a case example we've been working on that's currently in peer review and will hopefully be published imminently. This case example utilizes the topical spray formulation of desoxymetasone, but this drug product was only chosen because there was rare Q2 and IVPT data available in the public literature, which allows us to develop a model. It's not intended to represent a real pathway for generic development for this product. Because it's a steroid, that pathway would be a vasoconstrictor assay. You can see on the right we have the Q2 of topical spray, which consists of around 31% isopropyl alcohol. So from this we can reasonably conclude the tertiary composition, assuming that IPA evaporates rapidly, and we can predict the rate of evaporation in the simulator. There was no information available on Q3 properties for this formulation, and the aim of this study was to examine sensitivity to these properties under various conditions. So when looking to develop a model for this formulation, from the Q2 we can estimate the specific gravity of the mixture. We can also infer the drying rate and amount. We can parameterize the model with this information. However, there are a few important parameters that we don't know, such as solubility, for which we're most interested in the solubility in the tertiary formulation, and rheology. We can use the IVPT data, which was available in the patent, to optimize and verify the model. You can see the final observed versus simulated IVPT data on the right. Using the developed model, in this work we looked at various aspects of a theoretical virtual bioequivalence study. We looked at study power and the number of subjects required, the effective physiology, for example, would it make a difference if healthy or diseased patients were used in the study. We also looked at dose selection, which in this final slide I'm going to show you. The IVPT study that I showed you on the previous slide used a dose of 10 microliters per centimeter squared, which represents about 3.5 sprays per centimeter squared for this product. And this may not be representative of the clinical use. Therefore, we investigated the effect of different dose levels, which may be more clinically relevant on the outcome of the bioequivalence study. The outcome was quite interesting. If we look at the higher doses, for example, the same dose used in the IVPT study, there was some sensitivity to viscosity, albeit we do have to push to a pretty high value before the test product becomes bioinequivalent. However, interestingly, if we reduce the dose to something that may be more clinically relevant, such as 1 spray per 2 or 10 centimeters squared, the sensitivity to this parameter all but disappears. If we now look at the solubility, which is considerably more sensitive, and therefore we only need to modify it by around 50% before the test product becomes bioinequivalent, which, by the way, might be an interesting thing to know going into the development process. However, again, for the lower doses, the sensitivity was reduced significantly. This is particularly interesting for potent compounds like steroids or retinoids, where it might not be practically possible to apply them at a clinically relevant dose level in an IVPT study and be able to quantify them. This example shows an area where modeling can be used to fill in the gaps and limitations of such studies, and also perhaps to inform dose selection and study design of the bioequivalence studies. Thank you. I look forward to any questions and participating in the panel discussions.
Hello, and today I'm going to talk about how we're utilizing PBPK modeling to aid the development of inhalation platform products, showing an in-progress case study of a solution phase pressurized metered dose inhaler. I'm going to briefly cover the generalized approach, the data that informs the model, and talk through the model itself and close on how we hope to leverage this data as the program progresses. I know we're all likely familiar with the traditional bioequivalence paradigm, but it's worth reiterating the value of alternative approaches. For inhalation platform products, we typically establish sameness in composition, Q1, Q2, and of course in device and the platform operating principle, human factors, and critical component geometries. There's a suite of bioequivalence assessments in vitro, single actuation content and aerodynamic performance as determined by APSD assessment at a range of testing flow rates, to name the most crucial. From a safety perspective, there's PK equivalence, but for orally inhaled products for which the intended site of action is within the lung, The quantification of analyte from plasma is of course downstream of the intended delivery site, and this is one of the reasons that it's challenging to infer that PK is indicative of therapeutic equivalence. Which brings us to clinical endpoint studies themselves. In the case of respiratory products, typically assessing changes in forced expiratory volume, i.e. how much air a person can exhale during a forced breath, the argument that this is not a sensitive measure to discriminate closely related products is a moot point, but the real challenge is that these studies add around a year to the development timeline and can cost somewhere in the region of $20 million. Any methodologies, therefore, that can support increased success in any of the bioequivalence assessments or provide a scientifically justified alternative are of great value. Alternative approaches for orally inhaled inhalation products can be crudely summarized into three aspects: what we deliver, where it goes to in the lung, and what happens when it reaches its destination. And it's the latter 2 points where mechanistic modeling is most valuable. In defining what we deliver, this pertains to elucidating the structural components of both the formulation and the aerosol, and the concept of Q3 or arrangement of matter is helpfully introduced here. To understand where in the lung the dose is delivered, we utilize testing methods more aligned with patient usage, realistic APSD, leveraging more realistic throat geometries, and patient-simulated breath profiles. This data can then be fed into regional distribution modeling to derive where in the lung the inhaled dose ends up. For probing the fate of inhaled particulate, an increasingly common approach is dissolution methods, and this data is useful along with the regional distribution modeling in the PBPK model to predict PK performance. The end goal for us is predominantly to facilitate robust product development, increase our understanding of the progenitor product, and increase our likelihood of success in the clinic, i.e. right first time pivotal PK, and an almost secondary goal, the holy grail, a clinical endpoint waiver. The case study I'm going to talk through today is generic of a solution phase PMDI containing a single, highly soluble API. The test product is Q1, Q2 equivalent and comparable for all critical device parameters. I'm a materials scientist by training, and I entered the realm of modeling by first assessing, or rather trying to assess, formulation and aerosol microstructure. But in this case, there is no structure at the formulation level given it's a solution, and as the API is highly soluble, aerosol structure, investigating drug residual particulate, is perhaps less relevant than, for example, a complex dry powder product with multiple insoluble APIs. As such, I'm not going to dwell too much on the structural elements. For data feeding into the distribution modeling, in this instance, we've taken a range of simulated breath profiles, slow, fast, and medium COPD, and a healthy patient profile from the general literature. PMDIs are not dependent upon inspiratory flow capability for performance, nor do they generally offer much resistance. Even the severe disease cohort can achieve reasonably high peak inspiratory flow. But by necessity, due to the operational limits of the NGI, breath profiles are modified. This is because the mixing inlet for the NGI can't really accommodate testing flow rates above 100 litres per minute. We've also leveraged a range of throat geometries, the OPC, derived from MRI imaging of 90 different throat geometries in an attempt to cover the full potential patient cohort. The combination of breath and throat geometries is leveraged to try and cover the entirety of the patient population, but it's important to note in this example, we're using a fixed lung morphology, which is a limitation of the approach. For dissolution to aid the PK modeling output, the method we use is adapted from the standard paddle over disc, with dose and media volume to approximate a conservative estimate of epithelial lining fluid volume. An aerosol dose is collected post-anatomical throat, and the substrate is then transferred into dissolution apparatus. The dissolution of API in this example is very rapid, with over 90% recovered at the first sample interval. This indicates that in this case, dissolution is probably too rapid to be meaningfully discriminatory. I've included an example in the bottom corner here, leveraging the same method with a different product containing a slowly dissolving API, where we're able to show differential rate of dissolution dependent upon batch history, just to demonstrate this is less a factor of the method and more of the API properties. And on to the reason many of us are gathered here. For our deposition modeling, we're using Mimeticos Preludium from Amace Consulting in Sweden. This encompasses a lung model factoring number of airways, airway length, diameter, branching angle, average gravitational angle, and volume at defined capacity. The ventilation aspect includes non-steady state inhalation flow, breath hold, exhalation flow, and inhalation and exhalation volume. The transport aspect then is a filter in series model which takes the average dimensions of an airway region at the time the aerosol bolus is present in that region, which is subsequently used for deposition efficiency calculations, and these equations link deposition efficiency in each airway region to airway dimensions, residence time, particle size, and air velocity, and this is characterized by the realistic APSD data, more specifically MMAD. Output is then expressed as percentage deposition in the mouth and throat, the tracheobronchial region, bronchiolar and alveolar interstitial regions. This is the regional distribution data derived from realistic APST testing with multiple throat geometries and inhalation profiles, including the standard USB setup for comparison defined previously. And in this example, there's 120 NGIs for 3 reference products. The primary take-home is that for this product platform, distribution appears to be predominantly driven by throat geometry, more so than inspiratory flow capability, which is not too surprising given this is a PMDI. So EMACE have worked to verify their deposition modeling by comparing simulations to scintigraphy data, in this case tidal breathing of radio-labeled insoluble particles. The graphs illustrate deposition region as a function of aerodynamic diameter, and for fast on the left and slow on the right clearance components. To address potential differences in lung morphology, preludium predictions were made for 3 different lung model anatomies, including a disease model restricting various regions by approximately 25%. The 3 preludium models cover the scintigraphy data well, with the unconstricted model approximately centralized. This is the red line in the middle. It's of note that this verification is based on a dry powder inhaler drug delivery, and I'm talking about a solution phase product, but we can argue that this is applicable to most delivery systems, including metered dose inhalers, since it's only concerning the computational code for thoracic deposition calculations used within the model. The deposition model equations are agnostic, therefore, to the composition of the particles or droplets, and the only information pertinent to the model's deposition calculations with respect to aerosol properties are MMAD or GSD derived from the realistic APSD data. This is a useful approximation because once the particles or droplets have passed the otopharynx, before that, complex dynamic interactions between the aerosol and mouth and throat are dependent on a number of other factors that are challenging to model. To counter this, we've used realistic APSD with mouth-throat replicas and inhalation profiles to inform the deposition model. We are aiming to establish that PK is affected by changes in regional deposition and infer that PK parameters can be indicative of equivalence in local delivery to the lung. So this is what the PK model looks like. It is informed by drug-independent parameters, the physiology of different regions of the lung, drug-dependent parameters, the molecular properties of the drug in question, solubility, permeability, and tissue binding propensity, and of course product performance, in this case realistic APSD in various test scenarios, which then dictates regional distribution of the delivered dose. We have deposition in each region in the top, defined by the regional distribution data from APSD as mentioned, dissolution of the deposited dose, permeation of the solute over the epithelium to or from the lung tissue, and then exchange of the compound between systemic circulation and lung tissue by the perfusing blood. There's also an aspect of oral bioavailability and gastrointestinal absorption, which is drug moiety dependent, of course. Our output is simulation of the rate and extent of pulmonary absorption, or Cmax and AUC. Here is some of the API properties informing the model, reiterating the high solubility mentioned. It's important to note that this list isn't the full extent of parameters, but just rather an illustration. On to output of the simulation. The model shows generally good alignment with observed in vivo data, but underestimates uptake at initial time points. This could be a factor of epithelial barrier function in that the lung expresses more heterogeneity than the model accounts for. It could also be disease-related, but rapid uptake of this API is observed both in COPD patients as well as healthy volunteers. Paracellular uptake is highly dependent on lung barrier integrity, impacted by smoking, exercise, and a disease state, for example. We additionally tested the response of the model to dissolution by changing the dissolution model from assuming that the API was deposited as a solid on ELF to assuming a deposited solution. We saw no observable difference in the simulated PK output, further supporting the position that dissolution is not a critical product attribute in this example. When we leverage this model to plot predicted PK from all of our RAPSD assessments, we can see the model is sensitive to changes in long dose with simulated Cmax and AUC well correlated to in vitro long dose. Moreover, the test and reference products T and R in this example are well aligned, but the reference model is only informed by variation in the RAPSD setup, that's the breath and mouth-throat geometries, but the test output is informed by this as well as changes in aerosol performance by design. So we're in the position where we strongly believe that dose and dose deposition are critical and likely predictive of PK performance. So now we're at a critical juncture. We aim to validate the model by conducting PK assessment of the model test batches, varying long dose, so delivering equivalent total dose, but increasing and decreasing fine particle dose in the test batches. This will allow us to ratify model accuracy and establish if PK is affected by changes in regional deposition as we postulate, and infer that PK can inform equivalence in local drug delivery to the lung. It's important to note that the model in the model output is only part of the puzzle and not the entirety. There's more to be said about the identity of the delivered dose, but this can be shared another time. What this will be valuable for is as a development aid. With feedback from the PK assessment, we'll be in a position where we can use the model to aid adjustments in product performance and to predict PK bioequivalence prior to pivotal PK. So in summary, I'm a fan. Mechanistic modeling can be used to demonstrate the impact of formulation changes on the systemic absorption of inhaled products. In this example, we varied long dose both by formulation design with the modification of a volatile formulation component in solution and by variant test setup. All that remains is to verify this output clinically, a small hurdle, I say, tongue firmly in cheek. Regional long deposition is sensitive to changes in delivered dose, and PK is in turn sensitive to changes in regional deposition. So Preludium and modeling in general is a valuable tool for probing the sensitivity of PK to variations in product performance. Consequently, we believe that modeling is an invaluable tool to aid formulation and product design for inhalation platform products. We envisage these tools being incredibly valuable for ensuring right first time pivotal PK, defining appropriate clinically relevant finished product CQAs, and hopefully sometime in the near future to provide an alternative to CE studies. I'd like to end on some questions that we've yet to effectively address. First off, how do we effectively model the diseased lung state? Much of the data I've shown here is from healthy lung, and there are limitations to the diseased lung model we can leverage, some rudimentary restrictions to various lung regions. Both of these models are based on fairly old source data, so updates in this round would be beneficial. Secondly, what further computational evidence can be provided to establish that regional deposition can reliably serve as a surrogate for regional absorption? Is scintigraphy the only way forward here? We've explored scintigraphy as an option, but many of the suggested approaches impact product performance too significantly to be viable. And specific to this case study perhaps, is dissolution in the case of highly soluble APIs a sufficient discriminator of product performance? And is this valuable in the context of modeling? Movement on a bioclassification system for inhalation compounds would be valuable here too. So this brings me to the end of the presentation, and hopefully I've provided a little insight into the way we are using mechanistic modeling, both as an aid to formulation design and as I mentioned, to increase our confidence in PK conduct and to provide a robust submission platform moving forward. Many thanks for listening.
Good morning, good afternoon, or good evening, everyone. My name is Maxime Merdy. I'm a senior scientist at Simulations Plus, and today we will discuss about ophthalmic drug products and how modeling and simulation approaches can help to perform interspecies predictions and therefore support drug product development and regulatory assessment. As you can see on this slide, the eyeball is a very complex organ that includes multiple tissues, all connected to other tissues. There are different flux that allow the drug to exchange between all of those tissues, and therefore, to investigate the drug absorption, distribution, and elimination from the eyeball is a challenge, especially in humans where it's difficult to obtain samples. To treat ophthalmic diseases, the pharmaceutical industry has developed numerous routes of administrations, whereas eye drops and topical administrations, and then different forms of injections where the drug can be administered directly into the aqueous humor, at the back of the eye, or inside the vitreous humor, depending on where the target is and depending on the drug properties. However, for all of those drug products, if we go back to the FDA's definition around complex generics, and in this definition, one of the characteristics that makes a complex generic is the route of administrations. And for all of the ophthalmic drug products, the route of administration is defined as complex, so therefore they belong to complex generics. So in this presentation, we will focus on topical formulations, and before we jump into the PPK model per se and how it has been validated and utilized nowadays, I want to go over the different options that we have nowadays to develop ophthalmic generic drug products. To assess the bioequivalence of those drug products, there is possibly an in vitro only options that is based on the characterization and the evaluation of the Q3 performances of the drug product. With this in vitro only option, there is no possibility to have any Q1 or Q2 differences in the drug product composition and in the amount of excipients included in the drug products. So this option has been utilized, but actually when we go to the product-specific guidances, we see that this option is available but only for the simplest formulations, as well as for some other drug products that will be also the simplest one. When we talk about the simplest formulations, we talk about the solutions. I will get back to this diagram in a few minutes. The other option is to utilize clinical study, either PK or PD endpoint studies. And with those studies, there can be difference in the drug product formulation and possible Q1, Q2, and or Q3 differences. Let's first look at the PK endpoint studies. During a surgery of the eyeball, whether it's for cataract, keratoplasty, vitrectomy, or other kind of surgery, what would be possible to do is to extract one sample, typically the aqueous humor, but it could be another ocular tissues, and we would have one concentration at a given time point per individual. So if we repeat this process around 30 times, sometimes more, sometimes less, and we also do that now for the test drug product, we can then compare the PK metrics for both test and reference drug product and do the bioequivalence calculation based on the observed local concentration in the human eyeball. However, this method has some caveats, including a relatively high variability. Typically, only one sample per patient is obtainable, which would imply a parallel design. There is also a potential effect of comorbidities and co-treatments that can impact the B evaluation. And finally, in very some rare cases, a crossover design is possible. So that's for the PK evaluations. Now, for the PD endpoint evaluations, what would be done is, for a given patient, we could make a pharmacodynamic measurements, whether it's the intraocular pressure, the inflammations, or other mechanisms, and I would refer to the product-specific guidances. And we would measure a specific effect during a given time period. Similar to a phase 3 clinical trial, we would perform this evaluation in a large number of subjects for the test and the reference drug product. And then we could compare those responses based on the PD effect, but we expect a high variability. Regarding PD endpoint studies, I would like to quote a sentence from a publication that was published by the FDA 3 years ago that talk about comparative clinical endpoint-based studies, and notice they are like costly and sometimes they are insensitive to formulation of those differences, especially when the drug exposure and the clinical response relationship is flat. Which means that the PD endpoint studies, on top of being very expensive and a strong limitation to the development of generic drug products for ophthalmic route of administrations, they also maybe not be the best to really evaluate formulation differences. So now that we've looked at the different current methods to evaluate bioequivalence, let's have a look at the situation of the US market regarding ophthalmic drug products. So when I did this analysis back before the summer, there were 7 kinds of dosage form using the ophthalmic route of administrations: solution, suspension, ointment, drug-eluting contact lens, emulsions, gels, and then implant and or inserts. For all of those dosage forms, and for all reference standard, in blue you have all of the reference standard that do not have a generic on the US market. In purple, you have the ones that have 1 generic on the US market, and in green, the drug that have more than 1 generic on the US market. And we see immediately that for solutions, most of the drugs have a generic, but only a third or around a third actually have more than 1 generic for the reference standard. For suspension, around 20% of the drug product have generics available from the US market. But what strikes me the most from this slide is the amount of blue and the numbers of reference standard that do not have any generic, or if they have, sometimes it's just 1 generic on the US market. And I am insisting on that numbers of on the US market because of another study that was also published by the FDA, where they look at the impact of the number of generic drug product on the market and the price for the US population. And we see that it's only when we have more than 1 generic, and actually more than 2 would be even better, that we start to see a very significant price in terms of a significant reduction in terms of price reduction. So we established that there are some limitations to the development of ophthalmic generic drug products. Let's have a look at the issue. And for those drug products for which the site of action is before the systemic circulation, there is a challenge to estimate the drug concentration at the site of action, and that is for both safety and efficacy reasons. One of the solutions that we have been working now with the FDA for more than 5 years is the utilizations of PPK model to predict that concentration of the site of action and to analyze what will be the formulation differences impact on that local concentration. On this slide, you can see the PPK model structures we have developed in collaboration with the FDA since 2014. And you can see the model integrates all of the tissues that compose the human eyeball, as well as the eyeball for preclinical species. As we know, the rabbit is the main preclinical species that is used to investigate the ophthalmic pharmacokinetic of drugs, whether injected or topical. The strategies nowadays is to combine PPK model and preclinical studies performed in rabbit or other species in order to extrapolate and predict the human exposure for a given drug product. Before we could perform the extrapolation between the rabbit and the human, we wanted to make sure the model in rabbit was able to predict formulation differences. So in this slide, I will present you how we perform the validation for ointment dosage form. As any new evolutions that we put into GastroPlus, we first did a deep dive into the literature to identify the equations, models, and data that would help us to implement this new capacity. We then identified fluoromethanol as ideal case studies, and we had data for ointment, suspension, and solution dosage form. And you can see here on this slide the data set that we use for model development. We then perform model validation by predicting aqueous humor concentration in 5 studies for ophthalmic suspensions, where there were differences in doses or characteristic of the dose, including the particle size. After the model was validated, we then focused on the ointments, and you can see that the model was able to predict the ointment dosage form. I would like to refer you to 3 publications that we have published over the last few years that showcase how the model has been developed and utilized. And in those presentations, the question that we answered were, how can a PPK model be used to accelerate the development of ophthalmic generic drugs? And then also, what can we learn using PPK model? And since 2020, during our last collaboration with the FDA, the question was, what can we do using PPK model? And we entered a collaboration that focused on a new PPK and pharmacodynamic modeling strategies for ophthalmic drug products to inform their regulatory decision making, where we focus strongly on the interspecies extrapolation. As part of this collaboration, we've just published our first article that focuses on the clinical ocular exposure extrapolation for ophthalmic solutions using PPK modeling and simulation. In this article, we propose a strategy to validate preclinical to clinical extrapolation using the OCAT model, where we start by developing the ocular model in a New Zealand rabbit or an albinos rabbit. So we don't have to account for the binding to melanin in the ocular tissues. And then once the model is validated in New Zealand, we can then validate the model in Dutch-belted rabbit by accounting for the binding to melanin. Once the model has been fully validated in both rabbit species, we can then predict the ocular PK in human. Using these strategies that we put together, we focused initially on ophthalmic solutions because they're the simplest dosage form. And we also focus on PK extrapolation only before having a look at pharmacodynamic. And we published 3 case studies for levofloxacin, moxifloxacin, and gatifloxacin, as they had numerous PK studies published. Focusing now on gatifloxacin, you can see on the left here all of the data we used for model development and model validation in rabbit. And you will see automatically that all of the studies are not like perfectly described, but overall, the model was able to describe the ocular exposure in most of the ocular tissues in rabbit following the administrations of gatifloxacin at different doses and dosing schedules. Then, by simply changing the physiological parameters, but by keeping the same all of the drug-specific parameters, we were able to perform all of those predictions in human of ocular exposure of gatifloxacin, either in cataract subjects, and you can see in red the aqueous humor exposure in all of those subjects using in different clinical trials. We had 12 in total. We could predict the exposure in cornea and splitting the cornea epithelium and stroma for this particular study in green. And we had also predictions in vitreous humor for patients undergoing vitrectomy, as well as conjunctiva in healthy subjects. And once again, you can see that overall, although there are some inter-studies variability, the model was able to predict the data relatively well. I mentioned 3 case studies. So actually, I wanted to show you briefly like all of the results on all of the studies and the corresponding predictions. And just by having a quick look at it, we can see that the PPK model could predict really well the clinical exposure in human for those 3 drugs, and that in different surgery type. So to conclude, B evaluation of ophthalmic drug products is a challenge, and it's currently limiting the access to affordable drugs to the US patients. PK and PD clinical studies present significant limitations for generic developments, and one of the solutions to overcome those challenges is to use ocular PPK models, as they present an interesting approach to support the regulatory assessment of complex generic drug products. The OCAP model demonstrated its ability to predict the clinical PK for ophthalmic solution once it has been validated based on rabbit data. Currently, more research is ongoing, focusing on more complex dosage form, and we hope to publish those results really soon. With that, I would like to acknowledge my colleague Farah and Viera, as well as our collaborators from the FDA, Ming Liang, Ross, Andrew, and Liang. Thank you very much, and I will see you in the roundtable. Have a good day.
Hello everyone, this is Professor Ducharme. Thank you for listening to this presentation on model-based B for long-acting injectables. This is a follow-up presentation from the one that we gave last year at the previous workshop. Today we will discuss the 2 main approaches for model-based BE, the in silico continuation, which will be the focus of the presentation, and the virtual BE approach. We will present examples of long-acting injectables and what a model-based BE program would look like. We'll go step by step through this program, and we will discuss validation and other issues. Model-based BE is useful for much more than long-acting injectables. We published a paper a few years ago where in table 2 of that paper, we list the different situations and products for which model-based BE could be very useful. Suffice it to say that model-based BE are good for a plethora of complex drug products, such as the liposomal products. Oncology products are administered via different or complicated dosing cycles, and also implants that can deliver the active ingredient over a very long time. In PK, normally, we like to do a healthy volunteer studies to a crossover where basically we pass on equivalence metrics using non-compartmental PK. Non-compartmental PK can also be called observed PK because what we're comparing is the observed Cmax after a single dose and the observed area under the curve. With long-acting injectables, because they most often have to be given to patients, it is very difficult to do this type of study design. First of all, once you start therapy, you cannot stop it. So a washout is theoretically not possible. And so because of that, parallel design are normally preferred, and then the sample size becomes really huge because it is driven by the inter-CV and not the intra. Steady-state study design are also very difficult to do with long-acting injectables because one needs to demonstrate the achievement of steady state. And for some products, that may take years to achieve. And so because of that, we also have the problem of having to do the study in a parallel fashion, and the huge sample size makes the study really not practical or feasible. In contrast, once we use a population PK approach, we can fit the data that we have. So the data that we have does not have to be after a single dose or does not have to be at steady state. It can be at any time. And so we can fit data on day 1, or we can fit data on dose number 2, dose 3, etc. Or we can fit a multitude of different doses. All this data for the test and the reference product can be fitted to a population PK model that we call PPK. And this population PK model will give us 2 different types of results. It will give us the fitted individual PK results that are going to be useful for the continuation approach. And it can give us the population prediction, which could be useful for the virtual BE approach. If we use the continuation approach, once we have the individual PK results, we can simply continue dosing in silico in the exact same patients. So we can decide to continue in silico. So here we have a washout, and after that we have a single dose administration of the test, and we have a single dose administration of the reference. Or you can decide to continue in silico to a steady state condition, or whichever condition that you would like. So a population PK analysis, again, gives you individual fitted results and gives you population predictions. In addition, it gives you a number that we call the residual variability. This is all that is not explained by the model. So for example, if the residual variability is 15%, that means that there is 15% in the exposure or concentrations that we're not explaining. Again, 2 main types of model-based BE. The continuation in silico of dosing to the exact same patients that we've already discussed. This is based on the individual fits, the individual fitting of all the different profiles. So the nice thing about this approach is that bioequivalence is actually based on actual clinical patient data. We are not using any simulated patients for this type of approach. The validation is also simpler, as it is very easy to prove or establish that the individual fits are appropriate versus what is observed. The downside is that in this approach, you need a clinical study sample size that will result in the power that you need, so 80 or 90%. You will not simulate any new patients. The virtual BE approach uses the population predictions of the population PK analysis. So for this approach, you need a PPK model that is validated for this purpose. So you have to show and you have to establish that those predictions are accurate and they're precise enough for what you want to do. In practice, this is very difficult, especially for bioequivalence, because normally we see bioequivalence at plus or minus 20%. But the positive aspect of a virtual BE approach is that your clinical study sample size can be much smaller than the one that you are simulating for the virtual BE. So you can do, for example, a small study in 12 or 20 subjects to validate again that the population PK model is good for the products that you will be comparing. And then you can do your virtual BE analysis. So simulate thousands of bioequivalent studies with, say, 200 virtual patients in each. This approach can be done with a population PK approach or with a PBPK model. On this table, you have two examples of long-acting injectables, the long-acting injectables of paliperidone Invega Sustenna, which is a once-a-month product, and Invega Trinza, which is given once every 3 months. In the middle part of the table, you would have what would be considered conventional study design, if you could do them, presenting examples for parallel and crossover design. Whatever is in red is basically what is not feasible from a practical or clinical point of view. So either the number of subjects, of patients, I should say, is impossible to achieve, or the duration of the study takes too much time. In contrast, on the right side of the table, you have examples of what model-based BE programs could look like if you're using a continuation approach. The sample size becomes much lower, depending if you use the scaled or the average bioequivalence approach. And the length of the study, in ideal cases, could be as short as 2 months. One thing that is important to mention is that a crossover study design is much, much preferred to a parallel one, simply because when you're fitting the data, the only difference between a test and a reference formulation should be in the rate and extent of exposure PK parameter. So how does the long-acting injectable release the formulation and the relative bioavailability? All of the systemic PK parameters should stay the same between the test and the reference. So because of this, the model and the fits become much more robust. So the study design, if you can do a two-way crossover with average bioequivalence approach, could look like this. You could use 2 sequences, TR and RT. If you want, you can do a 4-way crossover design where you would suggest to use the scaled average bioequivalence approach. You could use again, the 2 usual sequences for this. Now, in some cases, 1 dose will not be enough to characterize the PK of the long-acting injectable. Let me give you an example. If you have a long-acting injectable that is given every 28 days, and let's say that the release half-life of the active ingredient is 20 days or so, then that means that over 28 days of sampling, you're not even characterizing 2 half-lives of this release profile. So that would not be sufficient. So in those cases, you may need to give 2 successive doses. So then the design will look like TTRR, and the other sequence RRTT. And in some cases, you may need to give 3 successive doses. So it all depends on the product. Let's look at the program step by step. First of all, we need to have a population PK model. If we only want to use average bioequivalence approach, we are not going to replicate the test or the reference, then we need a population PK model that is defined in terms of its PK parameter means. We need inter-CV for all of the PK parameters, and we need a low residual variability. This is important. If we want to pass at plus or minus 20%, this residual variability cannot be 50%. It will simply be impossible to pass. So this residual variability needs to be at most similar to what we would want to pass on. If we want to use the scaled average bioequivalence, then we need to have a population PK model that also includes what we call inter-occasion variability, IOV. The problem is that in practice, most of the time, the population PK model that are presented in the literature are deficient for a model-based bioequivalence approach, either because the residual variability is much higher than this 20 to 25%, or the model does not include an inter-occasion variability. So this means that as a sponsor, you would have to create this model, make one that will be applicable for a model-based approach. So you may have to do a pilot study minimally where you're going to give the reference twice and then create the model. Once you have the model and you're thinking of a design, for example, this one, then you need to test this design and present to the US FDA that the design that you have selected should be successful and discriminative. How you do that? You take your model, you simulate thousands of studies with 2 types of generic product. One that is equivalent at 95%, one that is not equivalent at, let's say, 79.9999% or 80%. Now, the equivalent product, you will simulate studies with a certain power. So let's say I simulate studies with 80% power, then at the end, I should show that I get 80% power. I don't want to get lower power, and I don't want to get higher power than what I'm supposed to get. Then I'm going to look at the inequivalent product, and I need to show that my alpha error is less than 5%. If I cannot show that my design is successful and discriminative, then that means I need to change it. I need to improve it. Maybe I need to add another dose. But once I prove that it is successful, then this is where you're going to basically write this report, and you're going to submit everything to the US FDA. You're going to submit the results of this report. You're going to submit the synopsis of your pivotal study design, all of the analyses and validation plan, and then you request to have a product development meeting with the Office of Generic Drugs. In our experience with sponsors, the Office of Generic Drugs has been extremely useful and have provided excellent comments and suggestions. They're very open-minded, and they really do work with you. Once the plan is approved, then you will do the bioequivalence model-based. So you will fit all the individual data to the population PK model. You will get the individual PK parameters. You validate the individual results. The fit has to be perfect. So if you look at this graph here, the individual fitted data has to be perfectly on the line of identity versus the observed. And you can have also present a table in terms of exposure metrics of what you observed and what is fitted. There should be agreement. Once you validate, then you can continue in silico and basically establish if your product is bioequivalent or not. Now, the validation of the PPPK model needs to be fit for purpose. So it all depends on what needs to be addressed. If the clinical PK study needs to be discriminative, so it needs to be at plus or minus 20%, like it is for a normal bioequivalent study, then really all you can do in practical term, I believe, is the continuation in silico, because that's the only one where it's possible with the PPK model to have a residual variability less than 20 to 25%, and so you should be okay at establishing plus or minus 20%. But if more than plus or minus 20% is acceptable, so let's say 33 to 50, or even 50 or 75%, then it would be possible in practical terms to use the population prediction of the model or a PBPK model, because for these types of predictions, of population predictions, the uncertainty is much greater. And normally, it's very rare that you're going to be able to get a model that will enable you to achieve this plus or minus 20%. So again, when we do population PK analysis, we get three types of results. We get the individual fitted results, we get the population predicted results, and we get the residual variability. The first two sets here, or that is what is used for the continuation approach. The last two is what is used for the virtual BE. Note that in the prediction, population predictions, your predictions can be, relatively perfect, like in this graph here. It really follows the line of identity. But as you can see, compared to the individual fitted, the predictions are always more uncertain. Okay, and so that's why most of the time you cannot achieve proving equivalence at plus or minus 20% with the virtual BE approach. The current issues that we see is, first of all, that we have difficulty getting population PK model in the literature that is fit for purpose to those model base. So, IOV is missing, or residual variability is high, or they're not fitting what we need them to fit. And so most of the time, the sponsor would have to do a pilot study. And so that delays quite a lot these programs. And so one question that we have and that we would need to establish or prove is, would it be possible to use a model that is theoretical, so probably very close to the final one, and then do the pivotal study, and where we would validate or change the model just using the reference product. So we would only get the reference data. We would be blinded to the rest, and we would therefore improve, validate the model. And once we have done that, then we would get all of the data and we would conduct the pivotal assessment. So this is something that we're wondering. Obviously, this would shorten the length of the program a great deal. So in summary, we've seen that this is necessary for many long-acting injectable products, but it's also good for many others. There's two different types of model-based BE. We've gone step by step on what needs to be done. We've discussed how important it is to have this discussion with the US Food and Drug Administration, and hopefully you will conduct this model-based analysis and pass. So at this time, I thank you very much for your attention.